Okay. The reason I'm thinking about this is because what we're going to do here is we have a very good summary of, here's what I'm thinking. We always knew interferon caused lupus, not always, but starting 15 years ago. Interferon had something to do with, type 1 interferon had something to do with lupus. We always knew that lupus made DNA anti-nuclear antigens. That goes back 50 years or 40 or something. It goes back forever. So somehow interferon causes nuclear antigen. But what is interferon really supposed to do? It's supposed to get rid of viruses. It interferes with viral replication. And it also, we are the first one to discover this, it also enabled B cells to make anti-nuclear protein antibodies. So you see the situation here. There's lupus over here making interferon and making autoantibodies, DNA. But humans survive on Earth, and the other animals too, because interferon both blocks the virus, and we think it's an endogenous effect inside of B cells, enables them to make anti-NP antibodies. And it's very simple. So, and then so that's a very, life on Earth would be impossible if that process didn't exist. And as a side effect of that very important process, some people get lupus. But lupus is a small problem. It's a side effect of this very important single function of having interferon. Really, I guess it comes down to interferon trying to interfere with viruses. And then somewhere along the way, we should probably figure it out. Somewhere along the way, interferon, it said another way to make viruses or protect against a second infection. So it's a dual purpose. First, it's up there for the first time as an innate response. And at the same time, it's saying, I'm going to protect you again. So these guys, that's why we're here today. So all of us are here today because of this second process. Because you might get it once and you want to get over it, and then you don't want to get it again. So the second thing interferon decided to do was, I'm going to go inside of a B cell. I'm going to have them make nuclear protein antibodies, but I'm going to make it so that you can make nuclear protein antibodies to anything. And the other tricky thing, this is so interesting, is that B cells, we know, need a template to be selected. I always noticed this so long ago. B cells just don't come up and make any antibody. There'd be infinitely large number, and they got to focus on a limited thing. And they're there to make protect from viruses. How can they protect from a nuclear protein inside of a virus when that virus, it doesn't even exist yet in nature? It wants to protect humans from all viruses, past, present, and future. The only way, some grand design from somebody up there or wherever, said, I'm going to use, I'll tell you what, and I thought this a long time ago, the only biological material that looks like a virus, that's inside of us all the time, that I can use as a template is your own DNA and protein complexes, or RNP complex like anti-SM, which we have another grant floating around for anti-SM. So the grand design is you can make antibodies similar to anti-SM, but not pathogenic. But those antibodies and those B cells that are developed, those will also respond quickly to any virus, because any virus is, I heard this the other day, a virus is basically dead. It doesn't exist. There's nothing there. It's a little bit of DNA or RNA, or sometimes the other RNA it's kind of an antisense RNA. So those three possibilities, but they're all nucleic acid and a little bit of protein, nuclear protein, wrapped together and kind of protected by a dead bunch of proteins on the cell, on the surface of the virus that enables its entry, kind of protects it, packages it. The virus surface doesn't do anything to hurt you, but the stuff inside does. But it's all dead. It's like picking up a piece of DNA on the floor or the lab. It won't do anything. But when it gets inside of a cell, then it causes a problem. And of course, that's part of evolution too, because some of our genome comes from a virus. But the important thing is this thing what's up here now, the nuclear capsid protein antibody. And the way we make antibodies to that is because we are already conditioned by nature and selection to use interferon to make nuclear protein antibodies. And I think the interesting thing is it's all based on interferon, basically interferon beta, because that starts everything. And I think it's interesting that if you have a good interferon response to begin with, you will eliminate the virus right away. Then you won't make a good immune response. If you have a bad interferon to begin with, type 1 interferon, then you won't eliminate the virus very easily at the beginning. You know, it's inside your lung in this case. And you also won't generate a good immune response. So interferon is really the key to making all this, making your immune response work. And the thing, so the problem or the question we have, and this is the key to think about, is it appears for this virus, you don't make good antibodies. So we would say either something's wrong with this model. I thought that this model many years ago, that must be the case. But this virus is especially interesting because some people don't make good long-lasting antibodies. So what's wrong with this model for this virus? I mean, I think we make interferon, but maybe not. Maybe make too much interferon, it clears up. Maybe the virus is in the lung. Maybe that's the problem. It doesn't get all over. It doesn't get up here. Up here, it gets into the lung. So maybe there's something special about the lung and its inability to make good B cells. Or it could be something special about this RNA and protein of this virus that somehow this is not close enough to our own.
Yeah, probably when it's so infected, they're already in the lung epithelial cell. In that case, very difficult to get in. So I think the antibody response is probably good for reinfection. But for the primary, if it cannot clear, if it's too late, generated too late, the virus already infected inside the lung cell, that's too late.
Well, yeah, that's one possibility. But the other thing I was just going to add is maybe the virus has finally evolved so that it's far enough away from our anti-SM and RNP, which we can check this. You know, it's drifted so far away that now, you know, because it doesn't want to have us recognize it, and it wants to be able to reinfect us. It's out there dead, and the only way it's going to come to life is if it gets inside of us. So it finally, this virus has decided, okay, if I can get inside of that guy, if I don't kill him the first time, I'll get back inside of him, and I'll be able to easily do it. So I'll make my RNP so that he doesn't have a very good cross-reactivity at this stage. And he'll have a lot of trouble making anti-SM, you know, the capsid protein. So for some reason, the immune response to this virus.
Yeah, they definitely make very good anti-MP. So that's not the problem.
Yeah, right.
But I always thought it's very difficult to make exactly the neutralizing anti-spike. How could it be so precise? You make an antibody exactly will be to block how could that be? Yeah, it's very difficult.
Of course, that's what the grant's about. You know, we have the
but the
direct and indirect.
Really recognize that binding side. So yeah, that to me, I thought that's.
It doesn't have to recognize the binding side. As long as recognize surface protein, they can neutralize. The antibody can neutralize. You bind.
Yeah, but they call it the receptor binding domain. That's where the antibody recognizes that domain and blocks the spike protein interaction with the ACE.
Yeah, but binds is somewhere else on the spike protein. It might not interfere with its.
No, that's where it's neutralizing. That's the neutralization antibody. Yeah.
So that's what the grant's about, and that's what I've always puzzled about for years. Yeah. Okay, we make RNP so we don't get viruses because
so making MP is not the problem.
Right, but I always puzzled. Well, wait a minute. How does that help make neutralizing antibodies against the virus capsid, surface proteins on hepatitis or whatever, AIDS, whatever you want? You don't. It doesn't matter if you make NP, but it must anyway, because the immune system cannot. It can barely figure out what kind of nuclear protein the virus is going to have, but by looking inside of us, we don't have that. You know, but it definitely can't figure out what capsid proteins they have by looking inside of us. And that's how the B cells are made. They got to look inside of us and find something that looks like a virus. And they got as far as doing NP. And but how does that cross react with the S? And maybe this is difficult in this case. I don't know why this one's so difficult as opposed to other viruses. So anyway, but our grant is about that question. That's what it's about.
I will present what the grant is.
Oh, okay.
So,
But it's a very
let me go through so they can see what we're talking about.
It's a conceptually
Right
such a central question to life on Earth that I think it's worth working on.
Right. So let me go through there. Remember, there are 3 COVID-19 grant, biomedical grants. One is about instruments. So we got 3 grants submitted about this. And the funny thing is, all 3 grants had 3 different models. So, okay, this is the first grant we submitted. This is submitted in March even. So that means the first time we're submitting a COVID-19 grant, we quickly put together. This is the simplest model we came up with. So this is when COVID-19 infected the lung. This is just what happened in the lung. So as a single-stranded RNA virus, so the question was simple. For people who develop strong inflammatory response, so why would some people develop such a strong inflammation versus some people may have very good antiviral response? So I think we proposed that for individuals who can develop very good antiviral response are the one that somehow the single-stranded RNA virus trigger the TR7 pathway. Whereas for some individual, for some reason, I actually did not propose in what way this happened. But as I remember, in older individuals, they, instead of going through the TR7, the epithelial cell tend to go through the TR8 spawn, which is an NF-kappa B. And that NF-kappa B is an inflammation gene, it trigger. Instead of if it's TR7 IRF pathway, that's an antiviral gene. So unfortunately, some individuals did not go through this IRF.
She muted. She mute. We couldn't hear.
Somebody's muted.
Which mute by herself.
Can you unmute? Oh, wait a minute. I think I did it as the guy. You know,
Well, you did.
Yeah, because you made me.
You did.
I don't know. I was trying to mute me because there was an echo.
Ask.
Unmute audio.
Ask to unmute.
Oh.
Okay.
You mean I said that nobody heard me?
Yeah,
Nobody heard you.
well, just the last minute or so.
We just saw you speak.
I was trying to fiddle around it. There's an echo, and I didn't like that echo, so I was going to mute somebody wherever the echo is coming.
All right. So bottom line.
I have the power to mute people,
All right,
but
yes.
actually, I muted the wrong person.
All right. So that's the little 9 months grant. So we'll study if the people who develop severe disease, SARS symptom, did they somehow, with the COVID-19, although it's a single cell stranded RNA virus, the single strand can trigger both TR7 or 8, but in those individuals, for some reason, it's a TR8 pathway got stimulated. So that's an NF-kappa B mediated, and that's an inflammation. So it's not an antiviral response. If we can successfully gear that viral response into the TR7, that would be beneficial. So it's a very simple model we're testing. So that's the first COVID-19 grant. There was no immune cell we say we're gonna study here. It's just the lung epithelial cell.
The hypothesis is very good.
Yeah, it's very simple, straightforward. So, it doesn't, why it doesn't do it? Something, oh no, okay. All right, so, but that wasn't really what we want to do, because after all, we like to study the immune cell, not the virus. So when we submit our own COVID-19 grant with the VA Merit Review Award, this is a 2 years award. So when we start to prepare for that, we said, but we cannot just dream or come up with anything. We don't know anything. So we asked Mingao to help us to look at the COVID-19 B cell, single cell data. So this is data, somebody from China, they actually analyzed overall 8 healthy and 12 just early recover patients. So these are recover patients. Initially, the data said they have 8 samples, but when I look at it, 5 out of those 8, so they cluster into 2 different groups, and 5 of them really express very high level of the hemoglobin gene. So I thought maybe there's some red blood cell contamination there. So I say, Mingao, we don't want that. So she
That's in the healthy control.
in healthy control.
I mean, in case you're thinking, Sharon's probably thinking about that's an inflammatory marker, but actually that's not it. These are the healthy people were screwed up. It's kind of surprising, because you'd think that why would, you can imagine patients being difficult, but why can't they get good healthy people? But we have the same trouble getting good healthy people.
Right. So she removed those. So ended up just 3 healthy, 12 COVID-19 sample. And these are the results. So the upper one, these are the gene that's been upregulated in COVID-19 compared to healthy. So we didn't do any trick. This is total B cell. So deep down, I know that COVID-19, they have more switched B cell. So in a way, we are comparing a little bit apple versus orange. So because of course, they have more switched B cells. Right, so you can see IgG, IgG2 and 3, but there's some interferon signature gene. So this one, and I think the box one are all interferon gene, except this one. And then you look at the healthy, well, I should say this is always a funny one. So we call it RNP negative versus RNP positive lupus patient. Then you could see that I think the box one are the same gene. So you see they show similar gene that's upregulated between lupus and COVID-19. And so it means that the transcriptome are very similar between COVID-19 and lupus. But of course, if you think about it, so it's an antiviral gene upregulation, and that's the type 1 signature. So that's not a surprise. But I asked Mingao to do this pseudotime plot. So this is the way we really like to do. So we can look at the trajectory of B cell from the earlier stage. So stage is defined from switching from IgM to IgG. So you could see that the COVID-19 patient definitely higher expression of IgG. And then, so you could see that IL-4 is slightly lower in the COVID-19 compared to healthy. So the resting naive genes are lower in general. But the interferon signature gene, you can see here that they are all upregulated in the patient. And then this is the DN2 gene, and they are also upregulated. And so you can see that this is COVID-19 pattern versus healthy. And then you can look at the lupus. Sorry, the labeling got shifted. So this side is lupus, that's healthy. And you could see that the cell trajectory and the transcriptomics are very similar between lupus and COVID-19 sample. So we kind of use this data to show that we can do it, and to say that there's an increased type 1 signature in the COVID-19 B cell. And here's more data. Sorry.
some COVID-19 patient going to the
Right,
hospital.
right. Yes, yes. I got a background noise here. Is that annoying?
No,
No? All
nothing
right,
you can do about it.
right. So you can see that this is the way we like to do it. We develop this using IgM, IgD, and then IgD only, and then IgG to show the B cell progression from the earliest to the latest. So you could see, and this is healthy, this is COVID-19, but the bottom line, yes, they have more switched. So they are IgG positive. And then here is the interferon signature. So COVID-19 patient had increased MX1, IR7. So the TR7 pathway in the COVID-19 is upregulated at all stages. So you could see comparing. So it's not whether we only look at, so whether it's not because they have more IgG, but every single stages of B cell development, they have more of this interferon signature gene. So that kind of took care of the problem that there are more switched B cell. And here is looking at the lupus. So the same thing here. So we use the same way. This is the IgM, and then IgM, IgD, and the IgD, and then IgG. So red is IgG, and then you can see lupus patient has more IgG. And that's, of course, that's the situation. Here, this is, I forgot whether this is gated in any specific subset, but I'm 100% sure, doesn't matter which stage it is, they have the similar upregulation of similar genes, so IR7 and MX1. So they're really just quite identical. But one is a chronic overall stimulation, one is an acute viral stimulation. So here's the question, so that with those data in hand, we say, how do we build up a hypothesis based on what we know about lupus? So in the second grant, so the second VA grant, in fact, we struggle. I have to say we struggle. And we finally come up with this model. But of course, Sharon helped quite a bit too, because initially it was something like interferon beta stimulates the SARS-CoV-2, and then they develop this viral MP response, because that's definitely the MP pathway. The MP binds to the RNA, so MP RNA together signals through the TR7 that stimulate the IgG, and that develop antiviral MP antibody. So we can understand that. So we really struggle a lot. So but that's not the critical one. What does that mean, right? Sharon, you remember that, right?
Yeah.
Because we keep on twisting, we want to twist it. How are you going to explain that? But really, people care it's anti-spike.
Right.
How would that fit? So we struggle and come up with this model that the spike is a foreign antigen that doesn't signal through the viral, the TR7. It doesn't do that. So somehow we that this pathway acts through the CD86, because interferon can stimulate CD86, and then this acts through the TBX21, TBET, and these are DM2 B cell, and these B cells produce the anti-spike. So we have to kind of squeeze hard, and this is the model we propose. And of course, we submitted. That's submitted in April. So, and down deep, I know there's something not quite right, because Sharon said it, and Cappy said it too, because MP is inside the cell. Even though you got lots of MP circulating, we keep on saying, yeah, but there's a lot of MP circulating everywhere, in the urine, in the blood, but it says, but it's when it's shedding, it's encapsulated inside. So maybe anybody can bind to it, but when anybody binds to it, what's the function of that antibody? How would that prevent the infection? How does that work? So to us, at the end, we submit the grant, but as I asked a lot of questions, it doesn't make sense. So we don't know how the reviewer received this. We don't know. But just to submit that grant is very helpful because you submit it and you know that's not right. We just want to get it submitted. So with the R01, we said we cannot do that. That's not right. So with that, and here's a little bit, not what we're doing here. This is the COVID-19 T-cell transcriptome. So same time, I think everything happened at the same time. So a paper published about this, the CD4 transcriptome with a single cell. And as it turned out, the signature of T cell they propose based on the transcriptome is more related to Th17, IL-23 pathway. So with those information in hand, we came up with a third model now in the third grant submission, which is based on what we know. And we think this one might be the closest to the reality. And at least I feel it cannot be too off. It might not be exactly, but it should be really, really similar to how the immune system works to get the right antibody. And it in every angle, it fits. So it won't have you won't have a big question on why it doesn't make it. To me, there's no doesn't make sense such thing in this model. So in this model, this is based on our previously published data. The B cell has two types of B cell. The first kind of B cell, they produce type 1 interferon. Whereas the second kind of B cell, they are type 1 interferon responding B cell. So the producer, and based on our previous work in both mouse and human, the B cell that make type 1 or express type 1 interferon, they are not the responding B cell. So there are two different subsets of B cell. And it started in the even earliest transitional stage of B cell. So with that model in hand, we propose this new model. So it means that the NP is generated because of the B cell make interferon beta. And so this is the B cell that continues signal through the TR7, as you can imagine. So this is the NP reactive B cell that with the RNA, single cell RNA component, that kind of little bit NP antigen attached to it together, they stimulate the TR7 pathway, of course, because it's NP. It's an RNA component, so it signals through TR7, and these are the interferon producing B cell because it's interferon beta signal signal through TR7. TR7, they make type 1 interferon alpha. So these B cell make interferon. So remember, it's the NP reactive B cell make interferon. But there's a neighboring B cell, no such thing. They don't have anything to stimulate them. It's very difficult for them to survive without exogenous interferon. So these B cell, the purpose of the NP reactive B cell is not to make anti-NP. Really, its purpose is to make interferon. The anti-NP is just a product because they signal through the NP pathway. But their real purpose for these B cells is to make type 1 interferon so that they can stimulate interferon responding B cell. These are the B cells that really want to make them survive, go through the maturation stages until finally they encounter the T cell. So once they encounter the T cell takes over and they make the spike antigen. So yeah, but without this step, this won't happen. Then during the development, they die because B cell receptor signal by itself is not sufficient. So without the interferon.
Can I say something here about this? Because this is kind of this is a critical point. So you wonder what, nothing is as usual, everything has a purpose. There's nothing that just happens and doesn't do anything. So you might wonder, so what do these anti-BNP B cells do? B cells and the antibody do, because they are made. So by definition, they have a purpose. So we know what the purpose is. This was shown by Rothstein and that guy, the guy Schlomchek. They showed a long time ago that the antibodies that recognize NP, nuclear proteins, anti-SM and RP. This is in lupus, but it carries over here. Lupus is a byproduct of all this. So anyway, those antibodies bind to nuclear protein, and then what they do is they've showed in a model, we wrote a grant about this too, that they bind to the Fc receptor on the surface of a B cell, and then they get taken up by the endosome, and the TLR7, 8, and 9, but let's say TLR7 in the endosome is there. And now these antibodies that bind the nucleic acid bend over and stimulate the endosome inside that B cell. And now that B cell has two things. It has the antigen that it brought in, the nuclear antigen is stimulating it, and it has a TLR7 signal, so it makes interferon.
Yes, Darman, you are right.
Yeah, so that interferon, those B cells that do that, that's an interferon amplification loop. And in the case of lupus, that bottom pathway is the disease. They go on and that's not regulated well, and then they actually get anti-DNA that causes kidney disease and so forth. But that's not what it's for. It's for this, it's for viral reaction to virus. And I believe it must just be for amplification. And it doesn't have to be on a B cell, by the way. Their model was a B cell FC, but it could be on a PDC or a dendritic cell.
Well,
so that,
B cell is the best.
Dr. Can I
so
say something?
Yeah.
You see, so B cell, you said, the VMP, the MP receptor and the FC receptor, band, that right? The engulf the virus, go inside the B cell, and then there are two pathways. One way it can activate the toll-like receptor. Another way, B cell is antigen presenting cell, APC cell.
Right. Oh yes. You're right. Yes.
So it can present the antigen. That's one way to activate T cell. And another way, B cell simultaneously, activated by toll-like receptor, so B cell can activate. And then this T cell, the antigen presenting pathway can activate T cell. And then T cell further help the B cell to make the antibody.
No, you're absolutely right, Simon. I thought of this. This is what let me just finish. This is very important. I forgot about this. That's what I figured there was I had the direct and indirect model. What you described is what I considered to be the direct model. That is, you bind nuclear protein, which is in a gob, and some spike proteins tossed in there. So then that means nuclear protein or spike protein goes inside of the B cell. And then it processes spike protein.
That's right. That's right.
That is the APC. But then
that's right. Yes.
And then it comes back and helps a spike protein B cell.
That's right. That's how NP protein help a spike protein.
Right.
So NP protein, you generate NP antibody, but that's really not purpose. That's just a signature you can find. But it's not protective. But NP by this way, the B cell they present a spike protein to T cell, and then T cell got activated further help the B cell to make the antibody.
Right. So here's the thing, that's what I call. I think I said the wrong thing. That is the indirect approach. What happens there is the B cell has now spike protein that's gobbed up with this NP. NP is critical, but it binds and brings in the spike protein. Then it's APC. Turns on a CD4 T cell, then it helps.
Yes, NP.
And then now you got a T cell, but this means that the T cell is the first step, making a T cell that can recognize spike protein in CD4 T cell. And then hopefully that same T cell can find the B cell that's been stimulated by the spike protein. So the T cell is immunized first, and then it helps a B cell. That's what I call indirect or two-step mechanism. See, what I prefer there's another way to work this, and that's what this is. I think that immune system would not be so sloppy. So take the roundabout route, or maybe it does both ways. The other way is what's shown here, is that the spike protein actually.
This way is still the same way, because here it stimulates CD86. That's what we propose. So this
yes, but the spike is driving the B cell. Yeah. So what you say, Shinrin, is the intermediate here for IL-17. But so really, it's like that's one thing, but that requires that this B cell that acts as an antigen presenting cell. Now, like I said, this same immune complex could go to a PDC or a professional, a dendritic cell. So maybe what's left out of here, what's not here, is that these NP.
That's indirect way. That's
right.
So that's called.
Yeah, but
it means it has to pull in. It has to somehow see. No, it's not anti-spike. It's anti-NP, and you got to hope that nobody's shown this. I looked for papers on this. Nobody's really shown that there's a bunch of NP spike protein complexes, floating around that can drag the NP drags the spike into an APC to process the spike and then present that on fast 2 MHC. Now maybe somebody knows, but I looked specifically for that, and no papers on that. But anyway, this model.
But there's paper showing the people who have good T cell response, they have anti-NP.
Yeah,
this is the thing you can say, what? It doesn't, but this model makes that sense, right?
Right. So we can figure all this out. That's why I think the key is to, even though we have B cells here, the key is to do PBMCs, T and B cells together, and see how they're working together here. And use some some of the Libra-seq.
This model explains everything too, including why NP. I keep on saying anti-NP is an early response, but anti-spike takes more time to generate. So this one go fast. This is antigen stimulate B cell, make the NP. But this one needs to go through in the barrel, stimulate the anti-spike require the T cell, so that it takes longer.
Well, it's a small difference, but this model proposes that the spike antigen finds a B cell that can respond to spike antigen. Which as a first step. And that to me just that's more efficient. The other model is that T cells are made, and then they have to hunt around for a B cell that recognizes spike protein and permit it to help. And it may be that both work. The spike protein has to independently stimulate a B cell, and at the same time, a T cell that has been, that was stimulated to an APC that took up a gob of stuff, that includes spike protein, processed spike protein, and the T cell became permissive. So those two things both come together. And we have it here because that's where the IL-17, because we didn't put in the T cell here. So that being that complicated, you can see that there's room for error. There's a possibility that some people can't do one or the other or both or whatever it is. They don't process antigens right. So that may be why some people make short-term antibodies, because if the T cells aren't involved, you'll make antibodies, but they'll be short-lived.
Right. If they have very strong interferon but no IL-17, maybe it doesn't work either. So it's
we know that this first step is interferon receptor. We just call it that because we propose not only does it require all this other complication that everybody, immunology 101's level, but this model requires there's two subsets of B cells, which we're the only ones who discover and believe that. And you can see they're different in the naive stage and B cell class switch stage. And then we left out the T cell and the APC. So even this model is an oversimplification.
Right, but for grant purpose, we did not want to study T cell. Right. So we stopped there. We're just going to say
for grant purposes, but not for.
Right. But the reason I thought IL-17 makes a lot of sense here for spike, because this is a totally foreign antigen. I think I said that too many times. Right. That this thing doesn't signal through TL. None of the TLR. that spike antigen can stimulate. It's just not anything you recognize, and not an innate pathway can recognize. So you need something, it's not type 1, it's not type 2 interferon, but it's something that can enhance NF-kappa B, potentially can do it. That's why we think, yeah, maybe IL-17 makes sense here to stimulate anti-spike B cell development.
Well, it could be, it's interferon. There's only two things. It's interferon gamma or IL-17. But now you got the data, right?
Yeah. So that's the R1 we submitted, and it will be reviewed in 2 months. So we'll see.
No, but you got the IL-17 data.
Yeah, that's right. This is from the published COVID-19 data. They suggest the T cell pathway goes through TH17 pathway.
Which was not a result of their paper. They didn't propose it. They didn't.
It's just a description. So that's what they found. They don't know what that means.
Right. It shot in and out of their head because everybody says it must be interferon gamma. I mean, it's a virus. Interferon gamma, and we got the TBAT positive T cells, TH1s. Everybody thinks it must be them, but it isn't. TH17.
Yeah.
We propose, or maybe it's both.
So of course, so with this model, it looks so complicated. So the goal is we're gonna generate that the Libra-seq method, so we can isolate spike versus NP reactive B cell. Then we can look at their transcriptome. That was the goal. But that was the R1.
Oversimplified.
Right. All right, so with that, now we say, okay, this is all COVID-19. What are we gonna do with lupus then? Well, actually, so with that, you can come up with a almost identical model for lupus now. So here's the lupus we did with, I think we actually did 4 healthy, 2 patients. No, over the time we have 4 healthy, 4 lupus, but here I show 2 and 2. So, and this one time we isolate total B cell, CD4 we isolated, we further gated into TFH, and then we get this is a plasma B cell, and then we also isolated T1 B cell. So here and then we did the hashing. Sharon, you must have remembered. And Ping An was there too. So this is just a very simplistic